Did some work with a particular persistent strain of Listeria monocytogenes. And what we did was, well, some of the work was done before I actually started my PhD. So I'm kind of following up on some other work carried out by Dr. Edward Fox. He's in Cicero in Australia at the minute. So what he had done initially was he had looked at a persistent strain of Listeria monocytogenes, particularly its strain 6179. And what we did is we assessed the growth of the bacteria in both the absence and the presence of an industrial detergent. In this case, it's benzethonium chloride. And what we did was that we exposed the bacteria to a sublethal concentration of the detergent so that it wouldn't die, but that it would, so it wouldn't be killed. So in an industrial setting, the detergent would be used at very, very high concentrations to eradicate the bacteria. But we wanted to see, because in certain niches in the environment, it's possible that the bacteria could be exposed to lower concentrations of this detergent and not be killed. And so we wanted to look at, what the bacteria does to adapt to these sublethal concentrations of the detergent. And so we enriched messenger RNA from the two samples. So we had the first sample was grown in the absence of benzethonium chloride. So we just looked at the regular growth. And then we exposed it to 5 parts per million of benzethonium chloride and enriched the messenger RNA from this as well. And we sequenced all the messenger RNA. And what we did is we took the bacteria's own sequence genome, which is provided by Dr. Stefan Schmidt-Hesse. He recently sequenced all of this. And so we took the bacteria's own genome and we mapped the reads back to it. And what we could do with that then was we could compare the two sets of data. So we could generate lists of genes that were expressed in both the presence and absence of the detergent, and we could, assess the difference between the two sets to determine what genes were significantly upregulated when the bacteria was exposed to the detergent. So what we did find was, the main findings we got from it were that there were significant upregulations in chemotaxis and motility, so flagellar biosynthesis, and several other components of the chemotaxis pathway were heavily upregulated when the bacteria was exposed to detergent, which is an understandable reaction considering that it's more a fight or flight response that it was exposed to this detergent, so the first instinct was to move out of this environment. We also found that there was a significant upregulation in carbohydrate uptake, so again it's another natural response considering the number of pathways that were upregulated and the number that require these sugars as precursors. And in particular, we also found a significant upregulation in genes involved in cell wall reinforcement, and peptidoglycan biosynthesis. Again, it's a relatively understandable response, I suppose, in that when the bacteria was exposed to this benzethonium chloride, its natural response would be to regenerate its peptidoglycan, considering the action of the detergent would be to break down the cell wall. And so they're basically the findings we have on it. There was a previous study done where the messenger RNA was mapped to a different but highly similar genome because the 6179 genome was unavailable at the time. And a number of these observations were also made, but we did find that once you have the bacteria's own genome, naturally enough, it's going to be better to do a study with that, considering that we found other upregulations that weren't seen in the original study. And so we kind of conclude that it's better to, I suppose, have the bacteria's own genome, if where possible, to do this kind of work. So that's basically it. Okay.
Post the prize.
I gotta give him the post. Congratulations.